Hello everyone, it is a great honor to share with you our paper. Comammox Nitrospira act as key bacteria in weakly acidic soil via potential cobalamin sharing. The corresponding author of this paper is Professor Baolan Hu from Zhejiang University. Nitrification is an important process in the nitrogen cycle, with ammonia oxidation being the rate-limiting step. Ammonia oxidizing microorganisms are the primary functional microbes involved in this process. pH is a key factor driving the ecological niche differentiation of soil ammonia oxidizing microorganisms. Theoretically, low pH is detrimental to nitrification due to its higher proton concentration, which also limits the availability of free ammonia. Despite this, high nitrification rate can still be observed in acidic soils, which is attributed to the presence of acid-tolerant ammonia-oxidizing bacteria. The discovery of Comammox Nitrospira in 2015 changed the two-step nitrification theory, as it can oxidize ammonia to nitrate within a single cell. Comammox Nitrospira exhibits a markedly higher apparent ammonia affinity compared to traditional AOB and the majority of AOA. The high abundance of Comammox Nitrospira in an acid mine and the discovery of novel strain in a weakly acidic bioreactor, suggesting that Comammox Nitrospira may dominate ammonia oxidation in acidic conditions. To reveal the impact of pH on Comammox Nitrospira, we collected soil samples with pH values ranging from 4.4 to 9.7 in China. The samples were categorized into three groups based on pH. Group A, pH less than 6.5, Group B, pH between 6.5 and 7.5, and Group C, pH greater than 7.5. We employed various molecular biology techniques to validate our conclusions. First, we quantified the abundance and activity of ammonia oxidizers AMOA. Comammox Nitrospira AMOA displayed a distinct pH preference, with both abundance and activity peaking in Group A and demonstrating a significant negative correlation with pH. The activity of Comammox Nitrospira explains 70% of the variation in nitrification activity. Thus, Comammox Nitrospira is the most abundant and active ammonia-oxidizing bacterium in weakly acidic soils. We performed amplicon sequencing on all samples and found that Nitrospira was the core genus. We identified for ASVs as potential Comammox Nitrospira ASVs. The sum of the relative abundances of these PCASVs showed a strong correlation with pH. The high correlation between these PCASVs and the positive cohesion with other bacteria further emphasizes the importance of Comammox Nitrospira in bacterial cooperation. The network analysis showed that Comammox Nitrospira is a key species in the soil and engages in cooperative interactions with other bacteria. We further performed metagenomic sequencing on all the samples. After assembly and binning, we obtained 121 medium or high-quality mags, including 3 Comammox Nitrospira mags. Using the same approach as with the amplicon sequencing, we found that Comammox Nitrospira dominates at low pH and may promote bacterial cooperation in weakly acidic soil. We constructed a cell metabolic diagram for Comammox Nitrospira in soil. We observed nitrogen metabolic pathways commonly found in Comammox Nitrospira, such as urea metabolism and complete nitrification. To adapt to low pH, the Comammox Nitrospira mags also encoded proton pumps, which are widely used acid-resistant strata to expel protons. Additionally, the mags of Comammox Nitrospira contained 3 sets of genes encoding F-type atposes, suggesting that Comammox Nitrospira can expel excess protons under low pH conditions. We annotated the genes of each mag using KEGG to infer the metabolic potential of the obtained mags. We found an imbalance in the supply and demand of cobalamin in the soil, with more than 86.8% of the mags encoding at least one cobalamin-dependent gene. However, only Comammox Nitrospira and rare Anella mags contain complete cobalamin biosynthesis pathway. We also validated the findings at the contigs level. Overall, these findings suggest that Comammox Nitrospira might act as the key species in acidic soils and one of the main providers of cobalamin under low pH conditions. Given the limitations of amplicon and metagenomic approaches, we further validated the reliability of our findings using D-and-a-CIP technology. After 56 days of incubation, D-and-a-CIP results confirmed that Comammox Nitrospira is the primary ammonia oxidizer under acidic conditions and may contribute to cobalamin biosynthesis in acidic soil. This is our conclusion.